Catheter-associated urinary tract infections, frequently referred to as CORTIs, are some of the most common hospital-acquired infections. This can be attributed to bacteria's tendency to adhere to surfaces and form biofilms, which are inherently difficult to treat. The difficulty in treating biofilms is due to additional antimicrobial resistance mechanisms exhibited by bacteria in the biofilm compared to bacteria in the planktonic state. Standard antibiotic treatments can often be ineffective against resistive biofilms. Functionalizing the surface of urinary catheters with drug-loaded silica nanoparticles could help to overcome this problem. Silica nanoparticles offer a sustained and targeted release of antibiotics directly onto the site of infection. This can effectively kill biofilms or prevent one from forming in the first place. The release of drugs from silica nanoparticles can also be tuned to a desired rate through the use of different stimuli. Here, from left to right, we can see how the release of drug differs under static conditions, stirring, and sonication, which is effectively a rapid vibration.